Okay, I am going to get started. So again, welcome, hello. Today's webinar is the impact of participation in data and genetic research insights from Simons Searchlight and SFARI researchers. My name is Lauren Bauer. I am the Senior Director of SFARI Cohorts and Biorepositories at the Simons Foundation Autism Research Initiative, and I'll be hosting the session and moderating our panel today. You should be able to see and hear me on the screen. I won't be able to hear or see you, but we do want this session to be interactive, so please utilize the Q&A to be able to ask questions. We'll have a Q&A session at the end after our panel discussion. We welcome you to ask questions, but we do ask that you don't include any personal health information or PHI in your questions, just for your own privacy. To get started, I did want to give just a few-minute introduction into the Simons Foundation and SFARI to kind of help set the stage. The Simons Foundation is a nonprofit private foundation with a mission to advance the frontiers of research in mathematics and the basic sciences. It was founded in 1994 by Jim and Marilyn Simons, and the foundation has been a champion of basic science through grant funding, support for research, and public engagement. We believe that asking big questions and providing sustained support to researchers working to unravel the mysteries of the universe. The Simons Foundation makes grants in 4 areas: autism and neuroscience, mathematics and physical sciences, life sciences, and science, society, and culture. Our Flatiron Institute was opened in 2016 and conducts scientific research in-house, supporting teams of top computational scientists. We actively promote collaboration across physical and intellectual barriers through a pioneering grant-making approach and are committed to sharing of knowledge within the scientific community. The Simons Foundation provides research funding for autism and neurodevelopmental disorders through the Simons Foundation Autism Research Initiative, also known as SFARI, which is sort of how we'll refer to it here. And SFARI's mission is to improve the understanding, diagnosis, and treatment of autism spectrum disorders by funding innovative research of the highest quality and relevance. And Simons Searchlight is a program that's funded by SFARI. SFARI has large investments in research cohorts, gene discovery, and open science. SPARK is an autism research initiative that aims to recruit, engage, and retain a community of over 50,000 individuals with autism and their family members living in the United States. Participation in this cohort involves the contribution of medical and behavioral information, mailing in saliva for genetic analysis, the potential option to have genetic findings related to autism returned, and the option to be recontacted to participate in future research studies. Simons Searchlight started in 2011 and aims to better understand rare genetic neurodevelopmental conditions. Simons Searchlight is an international research program that gathers family, medical, developmental, and behavioral information through online surveys and phone interviews with families and individuals. The Autism Inpatient Collection, or AIC, is a study that's performed by the Autism and Developmental Disorders Inpatient Research Collaborative, and it's funded by SFARI and the Nancy Lurie Marks Family Foundation. AIC began in 2013 and was designed to produce a collection of phenotypic and genetic data from children with clinical diagnosis of autism who've been admitted into one of 6 specialized inpatient child psychiatry units in the United States. Lastly, the Simons Simplex Collection is a core project and resource of SFARI. It achieved its primary goal to establish a permanent repository of genetic samples from 2,600 simplex families, each of which has one child affected with autism spectrum disorder and unaffected parents and siblings. Now I want to talk a little bit more about how these research cohorts interact and collaborate with each other. Many people who receive a genetic diagnosis from SPARK will be invited to participate in Simons Searchlight to continue their research journey. Participants in Simons Searchlight may have the option to provide a blood sample, which can be a valuable resource for the research community, and I'll talk a bit more about that in some of my later slides. And we also have Autism BrainNet, which is our biorepository of postmortem brain tissue made possible through a collaborative network of academic sites that collect, store, and distribute brain tissue. Simons Searchlight and Autism BrainNet share data and make the research easier. SFARI also funds outside researchers, which we'll learn a lot more about today from our panelists in our discussion shortly, and also makes the data from these cohorts available to the research community. This table shows some of the types of information that we collect from people with genetic variants in Simons Searchlight. We ask for a genetic lab report from the person who has the genetic condition, as well as other family members who had genetic testing done. We also ask questions about medical and developmental history and medication use. We use several online surveys to collect essential natural history information and understand how the genetic condition could affect daily life. Once that data is de-identified and curated by the Simons Searchlight team, we can grant access to that data for researchers through SFARI's online database portal called SFARI Base. As I mentioned earlier, donated blood can be used to make research resources, including cell lines, DNA samples, and induced pluripotent stem cell lines. Participants can donate blood that is shipped to and stored to our biobank called Sampled. Some samples are then sent to the New York Stem Cell Foundation for the creation of these iPSCs, and those resulting iPS cells are then sent back to our biobank where they can be distributed to researchers. iPS cells that we're making are made from Simons Searchlight participant blood donations, but they can also be made from other cells like skin cells. Certain blood cells are separated and treated in a special way called reprogramming, which tells them to become iPS cells, a process which takes about 6 to 9 months. I do want to point out that making iPS cells is not an experiment. We make iPS cells to create a resource for researchers to do specific experiments, and this can help save researchers time and money so they can focus on real experiments. iPS cells are special because we can force them to become the specific cells, a process called differentiation, that make up different tissues of the body, such as nerve cells that make up the brain, or liver cells, or heart cells. Those types of cells are typically not easily accessible from a living human, and iPSCs help researchers to study these types of cells from participants with certain genetic conditions. In addition to being able to understand how and why the cells of people with neurodevelopmental conditions function differently from those without a condition, the cells could also be used to find and test drug therapies. In addition to making the phenotypic and genetic data available, SFARI has a wide range of biospecimens that can be shared broadly with the research community. This includes tissue from autism BrainNet, DNA, plasma from the whole blood, iPSCs, and also animal models from mice, rat, and zebrafish. SFARI and Simons Searchlight want the information participants provide to contribute to ongoing scientific knowledge. This can include adding to the genetic reference guides such as ClinVar and gene reviews, which clinicians can reference, as well as articles in scientific journals and presentations at scientific conferences. Lastly, I want to talk about a way that we help ongoing research by letting participants know about new research opportunities outside of Simons Searchlight. So if groups are working with a particular researcher or researchers, they can apply to be part of this program called Research Match. Other scientists can ask to have specific surveys built into the system, and then that is sent to participants. Then the researchers can be sent the data, and it's also added to the larger Simons Searchlight repository to further build on the information that already exists. Research Match also lets participants know about in-person studies, which can include clinical trials. So with that sort of basic introduction into SFARI, Simons Foundation, and how we work with Searchlight, I do want to start with the introductions to our panelists. So first, I'll introduce Dr. Tom Frazier. He's a licensed clinical psychologist who received his PhD from Case Western Reserve University in 2004. He's a professor of psychology at John Carroll University, a national board member at Autism Speaks. And over the last 2 decades, Dr. Frazier has focused on improving assessment of autism spectrum disorder and associated neurodevelopmental genetic syndromes. Dr. Heike Rebholz is a neuroscientist who studies autism spectrum disorder from a basic clinical perspective. She was a postdoctoral associate at Rockefeller University, New York, and a research assistant professor at CUNY, New York, before starting at her lab at the Institut de Psychiatrie at Neurosciences in Paris. She's interested in phosphoproteomic changes in ASD and uses mouse models to determine the effects of dysregulated kinases on mouse behavior and underlying mechanisms. Dr. Julia Sommer is a molecular and developmental neurobiologist by training and is a senior scientist at SFARI. Together with the other SFARI scientists, she guides, evaluates, and manages SFARI's grant portfolio. Within SFARI's grants portfolio, she focuses on molecular and genetic studies of human neurodevelopment that make use of human-derived induced pluripotent stem cells. She's also the scientific lead for SFARI's iPSC resource, ensuring that SFARI's iPSC resource is well-positioned to support research that will accelerate our understanding of autism and related genetic neurodevelopmental disorders. Dr. Katina Calakos is a neuroscientist by training, currently on the Simons Searchlight team. She manages phenotypic data collection, focusing on medical, developmental, and behavioral surveys. She also develops the data summaries we share back with families and patient advocacy groups, such as quarterly reports and registry updates at genetic community conferences. Katina also works with the SFARI team to curate and describe the Searchlight data for distribution to external investigators doing research on Searchlight genetic conditions. So welcome to our four amazing panelists, and I'm just going to stop sharing my screen so we can get into our discussion. Okay, so after that introduction into SFARI, I do want to start with Julia, who has been at SFARI for a long time, and kind of understand a little bit more about how SFARI's evolved over the years and what impact it has had on advancing autism and rare genetic developmental research.
Hi. So I think SFARI's biggest contribution to autism research so far has probably been driving the discovery of genes linked to autism, and with that, the insight that rare genetic neurodevelopmental disorders are linked to autism. SFARI began supporting research on autism genetics around 2005, so quite some time ago. And back then, we basically didn't know very much, except that autism could run in families and had a high occurrence among identical twins, which suggested that genetics play a role. And this then led SFARI to start the Simons Simplex collection, which Lauren just mentioned in her intro, also referred to as SSC, that cohort of families in which one child is affected by autism, but other family members are unaffected. And then geneticists supported by SFARI used the genetic sequencing of SSC participants to discover the so-called de novo mutations that are only found in affected individuals. And that then revealed, really, that was a novelty at the time, that about 10% of individuals in the SSC with autism carry highly penetrant mutations in single genes or copy number variants. And over time, with the sequencing of additional cohorts, for example, SPARC, that Lauren also mentioned as a SFARI-supported cohort, it became clear that there aren't just a few genes linked to autism, but actually hundreds. And so this is when Simons Searchlight emerged, which set out to study individuals based on their genetic diagnosis rather than their autism diagnosis, which then helped us to see that not everyone with a variant in the same autism-linked gene actually has autism, and that there's also, in individuals with variants in different autism-linked genes, wide diversity of symptoms. But these individuals with mutations in different genes also share quite some symptoms. And that is especially true, of course, for individuals with variants in the same gene. And so based on this insight, SFARI now supports research delving into the molecular, cellular, developmental, and neural circuit changes caused by variants in many of those autism-linked genes. And this type of work often then involves studying animal or cellular models, And to support this type of research, we have funded the creation of iPS cells from Simons Searchlight participants.
Great, thank you so much, Julia. And could you also share some success stories or examples of researchers utilizing data or iPSC data from Simons Searchlight to make significant contributions in the field?
Yeah, so as I already hinted at a little bit, I think, because there's so many genes that we find to be linked to autism, SFARI is particularly interested in discovering if groups of genes linked to autism possibly share changes at the molecular or cellular level. And because we think this could lead to the discovery of drug targets that are shared across multiple neurodevelopmental disorders. And over the years, we've supported several studies looking into this idea. And I think one example would be work we funded Nail Nadif Kasri at Radboud University for. That's a good example. So the Kasri lab uses multi-electrode arrays, or MEAs, to study the activity of neurons derived from these iPS cells of individuals with autism related genetic disorders. And they initially started out, they had a nice study using iPSCs from 3 patients with Kleefstra syndrome. And each patient had a different variant in the Kleefstra gene, which is called EHMT1. And the interesting thing was, even though these 3 patients have different mutations, they found consistent changes in the neuronal activity and the molecular markers across all 3 individuals, which indicates that these 3 different genetic variants, even though they're in the same gene, share a mechanism. And that was really an important thing to show. And this type of approach can not only pinpoint disorder-specific neural network changes, but can also connect them with the underlying molecular changes, which could then be used to target them with therapeutics. And because we found this type of study really encouraging, we're now supporting Dr. Kasri to broaden this research to iPSC-derived neurons from individuals with variants in 18 different genes, all linked to autism. And the cells for this are mostly sourced from Simons Searchlight participants. And the goal here then is to uncover specific changes in neuronal activity for each gene and variant, but also to identify similarities among the different genes and possibly categorize groups of interconnected genes that may respond well to similar treatment approaches. So there's, again, this theme of trying to identify ways that we can use understanding cross-genetic disorders to inform therapeutic approaches. But at the same time, the research you'll be doing will also highlight the specific changes introduced by different genes, which then can also be used to develop very gene-targeted approaches, therapeutic approaches, as, for example, antisense oligonucleotide approaches.
Great, thank you. And could you also elaborate on the funding opportunities that are available through SFARI and how researchers can leverage them?
Sure. So SFARI usually provides funding for research projects through specific calls for proposals called requests for applications, or RFAs. Usually these funding opportunities, we try to bring together clinical research, genetics, and studies at the molecular cellular level to better understand neurodevelopmental disorders comprehensively. We have one type of RFA that's recurring pretty much every year. That's called the pilot RFA, and that supports innovative ideas that may be higher risk, but also potentially high reward. And the funding for such projects is $300,000 over 2 years. And in 2024, this will be announced in mid-April, and this RFA will be open to a wide range of research, including studies involving human subjects, as well as studying neural circuits, cellular and molecular investigations in animal models and iPS cells. SFARI is also currently developing a so-called targeted request for applications that's currently called the Development to Circuits RFA. It'll be announced this summer. And here we will focus on genes related to autism and neurodevelopmental disorders that play roles in very early brain development. And the goal for that RFA is to explore if changes during very early phases of brain development contribute to later alterations in neural connections and circuit activity, because that's actually currently not known. And the goal here is to gather detailed knowledge about the development of the brain influenced by these genetic factors, which we then hope could help identify the best times for interventions and the most effective approaches, possibly during different times of development.
Great. Thanks so much, Julia, for that introduction about SFARI and history. Now I want to delve into the crucial aspect of long-term data collection with Katina. Katina, could you elaborate on why we do longitudinal data collection and comment on how the team has addressed challenges that sometimes can come with ensuring ongoing longitudinal data collection?
Sure. Yeah, so when you contribute data, or when someone contributes data, we learn more about what is unique to that family, and also what's common across the genetic condition. And we also learn what is common across other genetic conditions that are included in Simons Searchlight, and that's a huge strength of ours, since we cover 180 rare genetic neurodevelopmental disorders. So while learning about the condition at one time point is important, it isn't sufficient in understanding the long term. So we want to know what the condition looks like at young ages, in adolescence, and in adulthood. So that's why providing those annual updates and contributing to longitudinal data collection is so important. But it is important, but we also recognize that there are challenges that can come with longitudinal data collection. So for one, it's hard because it requires time. So every year, we ask families to complete a subset of our surveys with the same questions. And so it could be hard to make time for that. It can also be a lot energetically, mentally, emotionally, to kind of think through what's affecting your family at that time. How have things changed since the last update? Maybe how have things not changed since the last update? And so we recognize that, and we understand that it can be burdensome to do this. And so to the point of, what are we doing to address these challenges? Really, in order to alleviate this burden, we try to make it as convenient as possible to fill it out every year. So for surveys where we can remind you of your previous answers, or we can make amendments to make the updates a little shorter, we do that where we can to reduce burden. Some surveys are just structured the way they are, certain standardized surveys. So we do appreciate the time you commit to filling those out yearly. And just as an example, with our annual medical history survey, like currently right now, we are amending it so that everything is entirely online, and you have essentially like a one-stop shop for all medical history reporting over time. We also know that the value of your research contributions, it can't be fully quantified, right? But we do want to acknowledge and show our appreciation for your contributions. So we do that with gift cards that can be redeemed online, and those are administered with baseline contributions as well as longitudinal data entries too. And then, finally, we also do our best to report back to you the value of your work by presenting data back in different kinds of summaries. So this can look like an individualized report that's available on the participant dashboard that can be used to learn more about like the specific participant as well as the genetic condition more broadly, like our seizure history survey report. It can look like these quarterly reports that we publish in a number of places, website, Facebook groups, et cetera, compiling medical history and then typically spotlights on different developmental and behavioral features of the condition. And then we also present it back at genetic community conferences throughout the year to different communities that are organizing these kinds of conferences, essentially overall to convey the value of your continued participation.
Great. Thank you so much, Katina. It's really very interesting to think about this in terms of long-term research participation. So I did want to turn it over to our two SFARI-funded investigators, Tom and Heike. Maybe we could start, Tom, with you to talk a little bit more about your current research efforts.
No, sure, thanks. That's great. I really appreciate you inviting me. I'm more on the clinical side, so it's been just amazing to work with SFARI because of the availability of these resources. The work that I'm doing right now is developing two different sets of tools. The first set is very similar to traditional questionnaires that parents oftentimes fill out about their children. They measure like different symptoms and skills, things like daily living skills, sleep difficulties, anxiety, ADHD, mood, etc. And we've built these measures in English. I did see a question in the chat wondering about some other locations. We're hoping to actually translate them into Spanish and other languages soon to make them available. So so that kind of work is ongoing. But I want to note that because of SFARI and the way that SFARI works, it was really great to have stakeholder inclusion in the process. And so we were trying to build measures that were much more appropriate and less burdensome for parents and caregivers, and ultimately more useful for patients. And we wanted them to be online measures to make it easier to collect longitudinal data and to eventually make it really clinically relevant so that we could be supplementing in-person assessment data. And then the second set of measures we built, which we're really excited about, were webcam collected measures that we collect directly from the patient and that look at their performance and sort of cognitive and behavioral features. And this tool uses some advances in artificial intelligence and computer science. And it's pretty exciting because, a lot of genetic syndrome patients are far away from these tertiary care centers. So it's not easy to get there. They might go once a year or maybe once every 2 years to meet with an expert to gather some in-person data. But with our tools, we hope that eventually we can collect the data in home and really supplement the traditional in-person data collections. And we just had a couple of publications, thanks to our data collections with SFARI, showing that our measures are reliable and have some good validity evidence. But we're also, again, working with SFARI on a new collection to try to expand that validity evidence and also to make sure that we're collecting additional genetic syndrome patients and autism cases and other NDD cases. So really that work just would not have been possible without SFARI. So again, thanks to SFARI, and just wanted to give people a sense of how, even on the clinical side, these tools are incredibly useful for our research and then ultimately to build towards clinical practice.
Great, thanks, Tom, for that overview. I know you were kind of talking about the resources. Was that, if you might just highlight on there, through Research Match that you were able to?
Yeah. So Research Match was huge for us. We were able to get a lot of our patients sourced, eligible patients who would meet our criteria. We could connect with them, figure out if they were interested, consent them. It just made the process so much easier. And as you can imagine, it's hard for the average researcher like me to identify neurodevelopmental genetic syndrome patients or even patients with autism that have particular characteristics or age ranges. And so Research Match was just so efficient at helping us to do those kinds of recruitments.
Great. And I know you kind of mentioned there might be some publications that have come out or will be coming out. Can you just highlight those a bit more on some of the specific facilitations of collaborations or breakthroughs?
Yeah, sure. So one of our we've had two recent publications, both in the American Journal of Medical Genetics, and the first one was on our traditional Informer report questionnaires. But if you look in that publication, we had groups like SYNGAP in there, P10, other groups were involved as well, like ADNP and STXBP1. And so these genetic syndrome groups, we just wouldn't have been able to get to them without SFARI and without Research Match. So, that's really a huge part of that publication. And then the second one was our webcam measures. And similarly, we had those same kinds of groups in there, again, recruited through SFARI. And we're already seeing differences between these groups on these measures. So it's pretty exciting that we can so quickly, using the tools that SFARI and Research Match and SFARI Base have available, to not only develop these measures, but test them in these groups and then compare them to the other data that's available through SFARI Base. I mean, that would have taken me a decade to do before, and we were able to do it in like 24 months. So
that's really, really wonderful and amazing work. All right, now I want to turn it over to Heike. If you wanted to tell us a bit more about your current research efforts, that would be great.
Yeah, thank you for letting me participate in this as well. So, you know, I've been working on a gene for many, many, many years. And now I'm working on a de novo mutation that causes autism disorder. So just like what Julia alluded or explained to during the process of research, Simons and you know, Searchlight and others have found all these new genes that we didn't know prior that they are involved in causing autism. So me working on you know, brain signaling, dopamine signaling, etc. Now I'm working on autism because we now know that the gene, a kinase that I've been working on, is causing autism. So I'm coming from this de novo mutation world, and Simons was already there at the time and had already a database for me to access. You know, I could speak to people there. Together with Simons people, we decided, because I'm a mouse model person, what kind of mouse models would be appropriate to generate first based on the patient, you know, mutation prevalence. So basically that's what I'm doing. I have different mouse models that represent different patient mutations, the ones that are mostly prevalent in that subcategory of autism. And I'm trying to understand also what Julia has said before. What are the common mechanisms that underlie these different variants, the gene variants, and what are the individual or more specific mechanisms that underlie, you know, the different mutations? So, and I'm doing that on mouse models. I'm studying their behavior, what is wrong behaviorally on these mouse models, and also biochemically. And I'm putting a lot of effort on proteomic analysis because I'm looking at a kinase, so proteomic analysis is important. So doing that with a Simons funding is going to be very helpful in understanding my subtype of the disorder. And can I tell you a success story? Well, we're just really starting. I mean, we have also a publication under review, hopefully it's coming out soon. We're planning to use, we have some pathways, some hotspots of, you know, mechanisms that are going wrong in our mouse models, for example, synaptic deficits. And so we want to use the Simons-produced iPS cells. They're still also being made because, as you know, they take a long time to be made and the controls need to be made, etc. So with these, we're going to test our hypothesis that we found in the mice, right? Are they also really underlying, or are they also part of the human cellular phenotypes? So that's very important. That's on the pipeline to be done very soon. And then another example that we came across not so long ago, we looked at inflammation markers in our mouse models of the disease, and again found that there are changes in the mouse model. And so now we want to validate again, is this inflammation, which has been shown, of course, in other mouse models of autism, but is that also you know, part of the human phenotype? So again, we can check that in the tissues that Simons has already collected in blood, etc. But we can also and aim, in collaboration with Simons and the patient advocacy group who are working together with Simons, to access, to reach out to patients and to ask them about, you know, for example, gut-based inflammation, because inflammation is not only prevalent in the brain, but also very much in the gut. So can we maybe access patient samples from the gut, i.e. fecal samples? Mm-hmm. And so this is something that we're planning to do and is already in the pipeline. I don't have a paper for that yet, but it's very, very exciting.
That is really exciting. So being able to use the SFARI resources in lots of different ways, it sounds like, for your work. Thank you both so much. That was really interesting to hear more about your work. I do want to add Katina and Julia into this conversation about the translational impact and clinical applications. Katina, if we could start with you, how are the findings from Simons Searchlight being translated into clinical applications for individuals with rare genetic neurodevelopmental disorders?
Yeah, Yeah, so Simons Searchlight data, it impacts clinical care now and clinical care in the future. So for the now piece, the data that's contributed is summarized back to participants, as I alluded to earlier. So this is, again, like the individual reports, the quarterly reports, the slide decks at conferences. And really the goal here is to provide news that you can use. So essentially insights that, you can take to understand the condition and to let the clinical care team know what others are experiencing in the community that the clinician or the clinical care team may not ever be able to see in their clinic, right? And then, additionally, the data also goes directly into the hands of researchers who are committed to better understanding this condition. So when researchers are downloading and analyzing the data, they can then draw insights about the condition and they can publish on that in the scientific literature. And then that unlocks, a whole new broader network of scientists and clinicians that can see that. So this can look like all kinds of things. This can look like a scientific paper, a clinical care guideline, a gene guide, a paper that links what happens in animal models or cell lines that have the genetic change with what happens in humans with the genetic change, right? So all of this combined means that a clinician who may have never seen a patient with the condition before can read through that literature, see what others are experiencing, and use that to inform clinical care in the now. But it also impacts clinical care in the future. So because we're collecting natural history data over time, and with the same structure over time, we can learn about the natural progression of the condition. And this can really directly impact the development of outcome measures for clinical trials and lead to better understanding of what's important in the community and what a therapeutic or intervention ideally would really need to address. So really, altogether, this could help design clinical trials down the road and pave way to therapeutics for the genetic condition.
Great, thank you so much. Julia, from your perspective, how does SFARI ensure the responsible and ethical sharing of data with both participants and the wider research community?
Yeah, that's a really important question. Hearing so much about data and biospecimens moving around across the world between researchers. So we mostly ensure privacy by providing researchers only with de-identified information about cohort participants. We also have all of those policies laid out in our researcher distribution agreement, which researchers have to adhere to in order to get even data access or biospecimens access. And this RDA outlines the conditions for data and biospecimen handling to prevent any, as I said, unwanted participant identification. For example, for the biospecimens, we don't allow researchers to directly transfer them to their other researcher friends, even if it's a neighbor's lab or so. Any request always has to come through SFARI, and any researcher using then the data or the biospecimens has to agree through our sharing practices. And yeah, as I said, that applies to data and biospecimens. And so I think that is a pretty rigorous process.
Great, thanks. That is really important. Another element about this is soliciting community feedback to be able to bolster research participation and engagement. Katina, going back to you, how does Simons Searchlight engage with and incorporate feedback from the rare genetic neurodevelopmental community in shaping research initiatives?
Yeah. So we do this in a few ways. So one way is our Community Advisory Council. So this council includes different stakeholders in the community, ranging from people with the genetic condition, caregivers to people with the genetic condition, patient advocacy group members and leaders, physicians, scientists from different institutions, all essentially to have diversity in thought and background, experience, and perspective. And this really helps us get insights about what is working well in Simons Searchlight and what we could be doing better. So just as an example, I'm part of a CAC working group on researcher outreach. And so talking about how we get the word out about our data to researchers. So I'm really looking forward to seeing what those working group members think about what we're doing and kind of how we can improve our researcher outreach efforts. We also get direct feedback from the community in all sorts of ways, right? That can be, emails to our coordinator inbox, social media posts, face-to-face interactions at family meetings and genetic community conferences, collaborations and discussions with other rare disease entities in the field, as well as going to scientific conferences where we can learn, more about best practices. So, we're really truly trying to get out there as much as possible and get the feedback that we need to constantly improve and enhance our research goals and processes.
Great, thank you. I'm sure that that relates to also being able to educate them, but could you be able to elaborate on how you're able to educate participants on the importance of their contributions and what kind of impact or potential impact it can have on the genetic research?
Yeah. So we really deeply value our participants' contributions to genetic research. So to ensure that people are informed and engaged, we provide those reports back that I've mentioned, highlighting key findings. We actively share discoveries at genetic conferences, and we really try to emphasize the real world implications of these findings. And, we also have our website. So this features summaries of scientific papers and publications that are focused on genes in the Simons Searchlight program, really in order to distill complex science into summaries that can be well understood, right, by participants and families. We have tons of educational materials, so data summaries, gene guides, other guides detailing what's important to the community, and all of this is available freely on our website and social media. So we really hope to give back to participants the value and insights from their research contributions. And we also collaborate with patient advocacy partners. So this really extends our outreach and ensures that participants receive information through trusted channels. And this really helps us foster community and shared purpose across stakeholders who are committed to understanding and treating these genetic conditions. So in essence, we believe that an informed and engaged community is just really vital for our success, and we're committed to transparent communication that empowers participants in advancing genetic research.
Great, thank you. And Julia, how does SFARI work to educate researchers about the value of our data and biospecimens available to them to complement their own research?
Yeah, I'd say we mostly do this via communication actively via our website or through meetings and workshops that we host, where we always try to highlight to participating researchers what's available, what data, what biospecimens, and also in personal communications that we have a lot with individual investigators. When we know what they work on and we know we have a resource that would help them with that work, then we'll tell them, and sometimes they actually don't know about it. So, but generally, we have our website SFARI.org that also contains publicly accessible lists of our biospecimens and iPSC resources, so researchers can go there and inform themselves. We also have on our website comprehensive information on the various cohorts that we talked about today, including Simons Searchlight. And I think anytime there is a new data release of Simons Searchlight, for example, that is highlighted on SFARI.org. So researchers know there's new data available, and this is how I can request it and get access to it. I think there's also on the Simons Searchlight website itself, there is another page for researchers and scientists where they can get more information on available data and biospecimens.
Great, thank you very much, Julia. So now I want to make sure that we have enough time to get to our Q&A and sort of wrap up by focusing a bit more on future directions. And so back to our investigators. Tom, maybe I'll start with you. What advice would you give to researchers who are new to utilizing SFARI resources in their projects?
Yeah, well, I mean, what works for me and what I would suggest is just work really closely with the team. They know their resources, they know the best ways to use them to facilitate recruitment, for example, and research match. And you know, there are lots of ways that you can fine-tune your process so that it's not only more effective, but also easier on the individuals who might participate. So I think that's one big thing for me. I would say the other thing for me is SFARI Base is an amazing resource, and it can actually decrease the amount of data you need to collect, and you don't have to be as redundant with data sometimes. So working with the team there to understand what's available and how that can supplement what you're planning to do, or at least can be something that you use to validate what you're doing is another really great option. So yeah, I would just really encourage researchers to work closely with the teams and think of it as a collaboration.
Great, thank you. And Heike, same question for you. What advice would you have? Oh, sorry, you're on mute.
Sorry. So what my experience from the very beginning was that Simons is not just a website, but it's really people who are really motivated and who are approachable. You send an email, you get stuck maybe ordering those IPS cells. You send an email and you get a person back with a name, and you can have an email exchange. I think that's wonderful. It's not a machine. It's really, there are people behind who are individuals who are motivated, who want to help you. Really, that's the feeling I've had many, many times. And yes, I also agree that the database has been very helpful for grant writing, very helpful for comparing your phenotypes or the data that exists not only on your little subtype of autism, but to bring it in the context. I mean, it's amazing. You have all these, it's like a comprehensive review that you don't find anywhere else. So I like that a lot. And becoming part of the researchers network for somebody like me who has started only a few years ago to become an autism person is truly a tremendous help, because I know who are the specialists. I can access them because I have the credibility of having worked with Simons. So this is amazing. And of course, the meetings that they host, so it's getting dark here in Paris, the meetings that they host are of the same spirit. Like, you know, it's a network. We're working together on this. We might work on different ASD subtypes, but we have a bigger project that's to bring the data together to answer big questions.
That's wonderful. Thank you so much for that. Julia, could you comment on what emerging technologies or methodologies that might be showing promise for advancing genetic research or understanding rare genetic neurodevelopmental disorder research?
Yeah, sure. I think it's become clear by now that I'm more the molecular cellular person. So I'm very excited about some advances in those areas. And particularly, I'm excited about those methods because I think they could help us to scale up research and understand many of those genes faster. So one method I'm particularly excited about is called cell painting. And that lets us explore many aspects of individual brain cells all at once. It makes it easier to understand each person's unique cell biology, again, using iPS cell-derived neurons, for example. And I think this could be a game changer in investigating the effects of different gene variants and develop actually personalized treatments, possibly. I'm also enthusiastic about a technique called single-cell long-read RNA sequencing. And this will allow researchers to understand how different forms of a gene, because it's more complicated. There's not just a gene, there's different forms that we call isoforms of a gene that are made in different cells. And so this can help us understand in which specific brain cells those different isoforms are made. And that could actually help us figure out if specific variants found in different people can affect different brain circuits, for example. So understand better the individual differences.
Great, thank you. And last kind of question on future for you too, Julia. How does SFARI anticipate addressing new research priorities and adapting to advances in the field of genetics and neurodevelopmental disorders?
Yeah, I mean, that's a really big question. I mean, in the end, I think the SFARI team, there's a bunch of scientists working here, not just me, like a molecular biologist, but there's others with more clinical expertise. And in general, we just try to stay in close touch with researchers from universities, companies, and then also the parent advocacy organizations focusing on specific genes. And I think this exchange with those groups really helps us to design funding opportunities that fill gaps that are not covered by other funding agencies. Although we also really like to work in collaboration with other funders. So we also talk to other funders to figure out how to adapt and how to change and how to complement each other and work together. And generally, we're really always open to feedback from researchers and family organizations. I guess, yeah, I think one recent example, for example, of a specific advance is maybe, and how we adapt to that with regards to funding, would be the development of wearable devices. So I think those devices now really allow to collect quite accurate physiological measurements from individuals. And so this now makes larger scale studies much more feasible, where before people had to come into a lab and get an EEG, now you could send them home with a device and you can scale that to many, many hundreds of people possibly. And I think we're, for example, now exploring this opportunity with a small pilot study of about 100 participants in the SPARK cohort to get a better understanding of sleep. So with wearable devices that can give you a readout about sleep. And I think, this is an example where we basically trying to adapt how we fund and what we fund in the SFARI cohorts to what new tools and methods are available.
Thanks, Julia, and thanks for mentioning this sleep study. I think it's really important and fascinating. So that is the end of our questions. I want to thank our panelists for all of the great input that they had, and I do want to take the last few minutes that we have here to open it up for questions from our attendees. I think some of our other folks have been able to take a look at some of the questions, and maybe some of the Q&As were answered. I think there's one in particular we thought might be helpful for any one of the panelists. Please feel free to speak up to help answer. And that's, what are the chances of conducting meaningful research on gene mutations such as SETD1A when the mutation is present in only 70 families worldwide? And any one of the panelists, feel free to chime in there.
Well, I've been working with groups that have similar prevalence rates, and I would say there's a lot of value in the possible work because, first of all, they're highly motivated communities, so you tend to get a large percentage of the 70, even if it's only 70. The other thing I would say is it's 70 now, but it might not be 70 forever, right? I mean, it's going to grow and grow. I mean, we saw a community I was working with that was only about 50 when I started working with them, and now they're like 350 or 400 people. So, there's a value in getting started on the 70 families now. And I would just say from a sort of more clinical and psychology perspective, 70 is plenty to start to understand some of the unique features of the phenotype. And I'm not a molecular person, but I could hear Heike and Julia saying something like, we can start to understand some of the molecular and cellular and neural systems level differences in those individuals as well, even with sample sizes of 30 and 40. And even when we start to think about maybe developing therapeutics, I think we're starting to move to a world now where we can start to look at therapeutics even in small groups of cases. And we've seen that in other genetic syndromes. So I'd actually be pretty optimistic about working with groups like that.
Fantastic, thanks.
I just wanted to chime in. I mean, this is truly the use of Simons Searchlight, right? To congregate those 70 families into a place where everybody can contribute their data and possibly biospecimens. And that's to get started, basically.
Yeah. Great. All right. Well, I do want to thank our wonderful panelists for joining today and sharing your expertise and insights with all of our attendees. We do invite everyone to stay engaged with SFARI by visiting our website at safari.org. You can also learn more about Simons Searchlight and the research program by visiting simonssearchlight.com. And if you have any questions about research participation or anything else we discussed today, you can also contact our team at coordinator@simonssearchlight.org. So thank you again to everyone and for taking the time to spend with us at lunchtime today and almost evening there in Paris. And we will hopefully speak to you all soon.
Thank you. Thanks for having us.
Thanks.